Hi, my name's Ryan, and today my peers and I are going to be doing a review of a formal figure by Kikuchi et al. From a letter titled Human iPS Cell-Derived Dopaminergic Neurons Function in a Primate Parkinson's Disease Model. I know it sounds really scary, but we'll just get into it in the next slide as you'll soon see. So here are some terms to know beforehand. Let's start with IPS. So IPS stands for Induced Pluripotent Cell. Essentially, they act as stem cells. However, compared to ESC cells, also known as embryonic stem cells, they are derived from fibroblast cells, which are typically seen in collagen or in connective tissue in humans or animals. And so the reason that we're using iPS cells instead of embryonic stem cells in this case is more of an ethical thing, simply because in embryonic stem cells, what you're doing is you're putting the tissue that you want to culture inside the egg. And some people have some ethical qualms about that, as you can kind of see, just because of the manner in which kind of the whole thing regarding eggs and how they're seen and perceived in society, and how we have this whole thing with abortion going on. But that's a different discussion entirely. Moving on, we have MPTP, which is a neurotoxin that we've derived, that essentially we use in the primate models in order to induce symptoms that are typically seen within Parkinson's disease. And then moving on from there, we have the substantia nigra, that is a region inside the midbrain, which is where a lot of dopamine or dopaminergic neurons, I should say, kind of originate from and kind of move around with. And just as a quick cover, dopaminergic, it sounds really scary, but all it really means is it's producing dopamine. And simply, what we do here in this process is we'll take a vehicle, which is our next term, which is essentially just a substance that we take whatever we want to put an agent into, and we use that essentially as a medium to transplant something from point A to point B. And then we'll also go over promoter, which is any chemical that really boosts the efficacy or efficiency of anything. Then that comes into how, like, we're using dopamine or trying to increase the dopamine concentration in this case of the primate models. And then last but not least, we have L-DOPA, which is also known as levodopa, which is used as a temporary treatment in Parkinson's disease, because what it does is it essentially increases the concentration of dopamine. And you'll see why this is actually important later in my next slide. So let's get into that. And then, so this is just a diagram showing the location of all these things, just so we don't get confused. And so this is just a cross section and through the brain. And so as you can see on my pointer here, we have the putamen, which my peer will get into later, and more importantly, substantia nigra, where everything kind of we're going to be talking about for now is happening. And so let's continue. And so the research objective of this letter was, so right now, we're talking about Parkinson's disease, and right now we don't have a cure for that. Like, yes, we do have L-DOPA or other dopamine promoters that can work as in the meantime, but there's currently no true cure for it, and that's where really the paradox lies. And so right now, what researchers are wondering is if we can use iPS cells, induced pluripotent cells, as this mechanism in order to produce normal levels of dopamine. And what they want to do in this case is they want to take it from patients, like human patients who are both healthy and PD patients, and we're going to transplant those into primate models, which are also in this case healthy primates, as well as primates with induced Parkinson's from the MPTP. And so obviously this is a scientific study. We want to apply this to a human model. We want to see if this works in the long term, most importantly, because short term, yes, it may do something, but more importantly is, can this be used as an effective cure for Parkinson's, is the most important things. And so how we're going to really measure that is based on motor function, because as you'll see in the next slide, that's really where Parkinson's really targets. And so let's go on to the next slide. And so this is a diagram I was referring to earlier, where essentially on the left side, you have the normal, you would have what was the normal striatum, and essentially all the dopaminergic neurons, also known as the tyrosine hydroxylase cells. And you see how it's taking, and so you see basically the dopamine concentration is really heavy right here and all throughout. However, in Parkinson's disease, it's very, very, very significantly affected in comparison. And so Parkinson's disease is a neurodegenerative disease that targets the nervous system and affects movement. Because essentially what you're doing is you have your dopamine concentration, which is very integral to be able to perform normal movements with your limbs. And it essentially prevents that normal motor function from happening. And so, and sometimes you might have, like, in the earlier stages, you might have like a tremor, but as it keeps going, is essentially what happens is that it'll start to affect the way your lungs expand, the way your muscles move, twitching in the eyes, etc. And on a cellular level, this is kind of echoed by a very much diminished level of dopamine. And so let's move on to the next slide. And so here we're just talking about our experiment design and flow. So as mentioned earlier, we're taking these iPS cells from these adult fibroblast cells, these connective tissue cells seen in animals, and we're essentially going to culture those into dopaminergic progenitors, which means essentially these precursor cells, really kind of the start of everything. But we do that by essentially just performing all these tests and using various chemicals in order to turn these connective cells into iPS cells. And then those iPS cells can specialize into the progenitor cells. And then once we take that, we transplant these into our cynomolgus monkeys. Oh, but that's on the next slide, sorry. And we do this via a vehicle, which we just talked about earlier. And then after that, my peers and I will get to this more important part of the statistics of it, which in our case of our figure, we did by doing MRI imaging and PET imaging inside the putamen. And then essentially we're just demarking the statistics of everything that happened within the 21 months of data research that they were doing. And so right here, as you can see in this column, are all these genetic markers. And essentially, this isn't as pertinent in our group compared to others, but it's just essentially all the genes and all the markers that were used within this experiment and all their effects on their own. But the culmination of these is what allowed us to transplant and culture these cells. But obviously, these look like small steps, but each of them takes time and a lot of resources in order to accomplish. And so the experimental rationale for this project was we're going to take these iPS cells and we're going to transplant them into cynomolgus monkeys, also known as Macaca fascicularis. And the reason why we use these organisms is because they have a genomic similarity that is very high with humans. I believe it's ranges somewhere from 95 to 99%. Either way, extremely likely, and therefore the prospects that we could gather from the data from this are exponential. Like, we could essentially derive there's a cure or not for Parkinson's from this data, if we adapt it to a human model, that is. And so also what they did, which I very much liked, was they used the PD models as well as the healthy models in both cases of humans and primates in order to gather data on the effectiveness of this vehicle and of the agent within it. And I think that will allow for greater potential of what we can do and what data we'll gather. And from here on, my peer will take it over. She'll introduce you to some new terminology and just take it from there. Thank you.
Hi guys, my name is Yesenia, and in the following slides, I'm going to be introducing our group's Figure 3, Parts I through L. Before we begin, I just wanted to recap that in the study, iPS cells were transplanted into primate models that had previously been treated with the neurotoxin MPTP, which causes PD-like symptoms. Once these symptoms were established for at least a period of 12 weeks, a monkey could be used for this study. These monkeys were separated into 3 groups according to treatment. So those that were treated with healthy patient cells were one group, those that were treated with PD patient cells were a second group, and then we have our vehicle group, which weren't treated with iPS cells. So the data in these following figures was divided and analyzed according to treatment group. So as Marian mentioned, after the initial iPS cell transplant into the primate models of the study, there were several methods that were used to observe and analyze the viability of these cells in the model's brains. Some of these methods included analysis of MRI and PET scans. So these figures were just one part of the visual and numerical representation of iPS cell survival and function in these primate models. As we go through each figure, I'll be mainly explaining versus analyzing which each figure illustrates. So for our first figure, figure 3I, we have MRI and PET scans of a representative monkey that was transplanted with iPS cells. So these images were taken beginning after the injection of MPTP and the establishment of PD symptoms, and then 1 week after iPS cells were transplanted, and then again every 3 months, and ongoing for 21 months. So the circle dotted lines represent where the putamen of the primate lies, and it's on both sides of the brain. So the bottom 2 rows depict PET scans that were used to visualize whether the grafted cells were actually functioning as dopaminergic neurons. So dopamine synthesis was measured by using a radioactive tracer, F-DOPA PET, and that's depicted in the second to last row. The expression of DAT, a midbrain dopaminergic neuron marker, was also monitored by using another tracer in the last row of the PET scans. So the warmer the color, the higher the expression of what was being analyzed. In our next figure, 3J, the information displayed by the visual scans was then used to create KI values, which is the average influx of dopamine into the brain's putamen. So these mean values are shown for every designated group in the study, and these were shown for over the course of 12 months. The gray dotted line represents an average KI value of 2 intact monkeys. So intact means this was before they were given MPTP, and then the onset of symptoms, and then given transplant. So these values just allow for the comparison, for value comparison. So in figure 3K, rather than mean KI values being graphed, the KI values for each primate were graphed for comparison. As in the previous figure, the dotted gray line is the average KI value of 2 intact monkeys. While the previous figure began at week 0 after the transplant, these values began with the scan of 2 intact monkeys before MPTP introduction, and then 3 months after MPTP introduction and PD symptom establishment. These scans were taken every 3 months after iPS cell transplant. The scans were continued for 18 months, and then the key on the side of the graph just gives you which monkeys were placed in what group. So for figure 3L, our last figure, we have a graph depicting a linear regression of KI values and TH cells for our 2 groups that received transplants. So TH cells are surviving transplanted cells that contain tyrosine hydroxylase for visualization and counting purposes. So this just confirms that the cells are active and alive. The vehicle group of this study was actually omitted from this regression due to low numbers. The blue dot designates the group who received transplants from healthy subjects, while the red dots represent the group who received transplants from subjects with PD. So after discussing these figures, our group reached some main conclusion and discussion points. So for data explanation, over time, KI values in transplanted monkeys increased, as did TH cell numbers. KI values for those in the vehicle group did not improve. So this data was actually used for the estimation of graft growth over the course of 24 months, and an equation was actually derived out of all this data. But our next presenter will discuss these conclusions with you. Thank you.
Hi, my name is George Demestihas, and today I'm going to be telling you about the conclusions that we were able to draw from figure 3, I through L. So in the journal, we received figure 3, I through L, and upon reading it and upon looking at the really good research that those scientists did, we are able to determine that grafted cells appear to be dopaminergic and able to grow in the brain, and this was indicated by 2 things. The first thing being the presence of tyrosine hydroxylase, and the second being simply graft growth. So the reason that the presence of tyrosine hydroxylase is promising for these progenitors to be dopaminergic is that if tyrosine hydroxylase is present, that means that dopamine synthesis could possibly be occurring. Now, tyrosine hydroxylase is an enzyme that converts the amino acid L-tyrosine into L-dopa, which is a precursor for dopamine. In addition to that, graft growth and volume of the graft rather was able to be observed visually through MR and PET scans. So if you look at the dotted lines that are on these brain scans, the dotted lines represent the average size of a putamen, and then obviously you have the midbrain in between those. You have week 1, 3 months, 6 months, 12 months, 18 months, and 21 months. You can see that the very first scan labeled MPTP, there is very little there's no graft growth, and there's very little tyrosine hydroxylase indicated, which would be expected. Now, you can see that over time and as time goes on, the presence of tyrosine hydroxylase both increases as well as the size of these grafted cells. Now, this is very promising because if tyrosine hydroxylase is being produced, then that means that dopamine synthesis is likely occurring. And in addition to that, if graft growth is occurring, then that means that these could potentially be used in order to replace those dopaminergic neurons that are lost in the subjects due to Parkinson's disease. In addition to that, researchers found that the Ki levels of transplant receiving monkeys reached 48% that of normal monkeys. So the Ki levels are basically the influx value of how much these grafts have grown. So what this is essentially saying is the grafts have grown to about 48% of the size of normal monkeys, which is good. I would speculate that with further observation, the grafts would continue to grow based on this research, but you can never be too sure. Now, as far as the professional critique and feedback, my group and I felt that the research presented in this journal was both accurate and can be repeated. However, there were some things that we would change or recommend. So obviously with research, the biggest thing is repeatability, and we feel that if they had shown more scans of multiple different monkeys rather than just the scans from one representative monkey, which was monkey number 1, we feel that it would really hit home the idea that this is a process, and that these experiments that these scientists are doing could actually be repeated and could actually give hope to those with Parkinson's disease, and that in the future there could be a possible treatment. So definitely would like to see more scans on those, just so we can see like the trend of growth over time. In addition to that, I think it would be a good idea for them to conduct this experiment in its complete form multiple times. And I think that this should be the case because if you can't repeat the experiment, then it's not really a valid experiment, and it doesn't have any scientific value except to know like, oh, we shouldn't do that anymore. So I definitely think that if they conducted this experiment at least twice, it would show that their methods and their process is consistent, and it would give a lot more credibility to what they're saying. Not saying they're not credible now, I'm just saying it would definitely be more compelling to show that you could do this over and over multiple times if you needed to. Thank you.